We will now take up 10 more questions which were asked in the zoology section. So let's start with the very first question over here. Here they are asking, the fruit fly has 8 chromosomes. This is the diploid number of the fruit fly in each of the cell. So the diploid number of fruit fly cells we have got over here is that it is 8. Now, during interphase of mitosis, if the number of chromosomes at the G1 phase is S, so we know that the chromosome number at G1 phase is equal to S over here. We know that the number of G1 chromosomes is diploid only. So, having 8 chromosomes is very much correct. Then, what would be the number of chromosomes after the S phase they are asking? Now, we know in the S phase what happens, the doubling of genetic material takes place. That means the chromosomes, they undergo the doubling and there is the presence of sister chromatids which is seen. Obviously, the chromosomes during the S phase, they are in the relaxed state, that is as a reticulum, that is as a chromatin reticle. So, what they are asking over here though is, what is the number of chromosomes after the S phase? The number of chromosomes, however, after the S phase, after the G1 phase, G2 phase, it remains the same only, that is the same diploid number. We have to understand one thing, that during the S phase, it is only the doubling of genetic material, that is the content is doubled. However, the number of chromosomes, it remains the same. So here, if they are asking the number of chromosomes remains same, we will go with the option 8 itself and C forms the correct option. So answer is C. A very simple and an interesting question. Now, let's come to a next question. Here they are asking if the adenine makes around 30% of the DNA molecule, what will be the percentage of thymine, guanine and cytosine in it? So now by studying the Chargaff rule, we know that the amount of adenine is always equal to the amount of thymine. So if adenine is equal to 30%, then definitely we can say that thymine is also equal to 30%. So adenine is also 30 and thymine is also 30. So now let's look at the option which is saying thymine as 30, and out of A, B, C and D, only A option goes with thymine as 30. So we can understand that answer is A, but then let's work up the other part of the question as well. They are asking the amount of guanine also. Now adenine 30, thymine 30, that means 60% of the DNA is already taken up out of 100. What is left behind is 100 minus 60, that is 40% of the DNA is left. And this 40% of DNA that is left, it is going to be segregated between guanine as well as cytosine, both of which are in equal quantity. That is also explained by Chargaff rule. So if we have to split the leftover 40 into equal parts, we will get guanine also 20%. And similarly, the cytosine will also be 20%. So now we have our values. Thymine 30%, guanine 20% and cytosine 20%. So we have come to our answer, that is the A option. Now let's see a next question. Here they are asking that which one of the following organism bears hollow and pneumatic long bones. So this is a feature of aves, that is, this is the feature of birds. So in all the options that is given below, we will have to find out the option which is having a bird in it. Macropus we know is kangaroo. So obviously this is not coming under aves, it is coming under mammalia. Then Ornithorhynchus is known as duck-billed platypus, which is again a egg-laying mammal. Then nephron is a vulture, so obviously it is coming under bird. And Hemidactylus is wall lizard, so which is coming under reptiles. This is again not the option. So the answer very clearly for this particular question is nephron. Now let's talk about another question. Here they are saying which is the only enzyme that has the capability to catalyze initiation, elongation and termination in the process of transcription in prokaryotes. So now what is transcription? First we'll have to understand this process. Transcription refers to the passage of information which is stored in DNA into the messenger RNA. So here what we have to do, we have to form a RNA molecule from the DNA molecule. As a result, the enzyme required over here will be a RNA polymerase. Why? Because RNA enzymes are formed. This information which is used for the making up of RNA molecule will be taken from DNA. So the enzyme used over here will be DNA dependent RNA polymerase. So now let's look at the options. Which of this is having the DNA dependent RNA polymerase, which is helping in all the three steps of the transcription? DNA ligase. DNA ligase is the enzyme which helps in ligating, joining the various nucleotides. So obviously this is not correct. Then DNase is the enzyme which is required for the breakdown of DNA. This is also not required. Then DNA dependent DNA polymerase is the enzyme which is required during the process of replication. But here we are talking about transcription. DNA dependent RNA polymerase, as I told, is the correct option that is required in the case of prokaryotes. So answer to this question will be our D option. Now let's talk about the other question. This is again a matching type of a question. Here we have to match the options which are given in list 1, that is Physalia, Limulus, Encyclostoma and Pinctada, with their common names which are given in list 2. So the common names given over here are pearl oyster, Portuguese man of war, living fossil and hookworm. So now let's start with option number 1. Physalia we know is Portuguese man of war. So A will be matched with 2. The option having A with 2 is only A and C, whereas both of these are not correct. So our option D as well as B can be cancelled out from here itself. Then Limulus is known as king crab, which is a living fossil. These are those organisms which have not undergone any change from a very long period of time. So Limulus is an example of a living fossil. B will be matched with 3. Here in both the options B is matched with 3. Then Encyclostoma is a hookworm. It is coming under Aschelminthes. So here C will be matched with 4 and we are just having one option, that is option number A, that is matching C with 4. Whereas this matching is wrong. Then Pinctada is the other name for, it is the scientific name for pearl oyster. So we will match D with 1 as correctly given in option number A and incorrect in option number C. So our answer to this question is option number A, which is given over here. Now let's talk about another question. So this is a very interesting question. Here they are asking which stage of the meiotic prophase shows terminalization of chiasmata as its distinctive feature. So now these are the various options of the prophase 1 of meiosis. They have given dikinesis, pachytene, leptotene and zygotene. So there are 5 stages in prophase 1 of meiosis. Those 5 are leptotene, zygotene, pachytene, diplotene and dikinesis. Leptotene refers to that stage where the condensation of the chromatin fibers is happening. So condensation of chromatin fibers is seen in leptotene. Obviously leptotene will not be the answer. Then zygotene is that particular stage where the homologous chromosomes are now going to come together. They are going to form an association that is known as synapses. So synapses is seen during the zygotene. So obviously zygotene will also not be the option. Then pachytene is that stage where crossing over is taking place, that is exchange of segments, that is non-sister chromatids are exchanging their segments. So crossing over is seen in the case of pachytene. So obviously this will also not be the option. Then in diplotin what happens? Now the homologous chromosomes, they separate from each other. They start separating. However, they remain associated at the positions where crossing over had taken place, and those positions are now known as chiasmata. So chiasmata formation will be seen during the fourth stage, that is the diplotin, right? And this chiasmata terminates in the next stage, in the last stage, that is known as dikinesis of prophase 1. So answer to this question will be dikinesis, that is option number A. Now let's see another question. Here they are asking Dobson units are used to measure thickness of. So this is a very general theoretical question. Dobson unit is used to measure the concentration of ozone in a particular area. So out of all these options, the answer ozone stands correct. Next, sphincter of Oddi is present at. So now what is sphincter of Oddi and where is it present? Is it present at the gastroesophageal junction, that is between the stomach and esophagus? Is it present at the junction of duodenum and jejunum, that is the parts of small intestine? Then is it present at the ileocecal junction, that is in the junction of the ileum and cecum, small intestine and large intestine? Or is it present at the junction of hepatopancreatic duct and the duodenum? So we can understand this question by looking at this figure. This is a picture. This C-shaped structure is duodenum. In the duodenum, we know that the secretions coming from the liver, that is the bile, coming from the common bile duct, as well as the secretions from the pancreas, which are coming from the main pancreatic duct. These both ducts, they join together to form a common duct known as the hepatopancreatic duct. This hepatopancreatic duct, it opens into the duodenum through the ampulla of Vater, and the regulation of this opening is done by a sphincter that is known as the sphincter of Oddi. So we can say that sphincter of Oddi is present at the junction of hepatopancreatic duct and the duodenum. So out of all the options, the answer number D holds true for this particular question. Now let's come to the next question. Here they are saying in a cross between a male and a female, both of which are heterozygotes for sickle cell anemia gene, what percentage of progeny will be diseased? So here the parents, they are heterozygous for the diseased allele. Now if we express sickle cell anemia as a normal allele for sickle cell anemia is represented as A, this is normal, whereas the diseased allele is represented as S. So if we have to show the heterozygous parents, we will show the heterozygous parents as HBA, HS, or the other parent is HA and HS. So this is parent number 1 and this is parent number 2. Now we know the diseased condition will be seen only during a recessive condition. So whenever there is HBS as well as HBS, that is the recessive condition is present, then only the diseased progeny will be witnessed. So now if we look over here, the gametes of one parent are written over here, HA and HS. Similarly, the gametes of the male parent are written over here, HA and HS. And now if we fuse them by the help of a Punnett square, we get first offspring, which is HA. That means this is going to be a normal type of an offspring, not showing sickle cell anemia. Then the offspring having HA and HS is going to be a heterozygous, but this type of a heterozygous individual does not express the disease. However, we can call it as a carrier. Why carrier? Because it can pass away the allele to its next generation. So HAHS over here and HAHS over here as well, both of them are carriers. And we get only one option, that is HSHS. This is going to be the diseased progeny. So if we count the whole percentage of all the children, we see that only 25% of children will be suffering from this disease. So out of all the options, that is 25, 100, 50 and 75, we will go with option number A, that is 25. Now, let's talk about another question over here. This says that a person with AB blood group are known as universal recipients. What is the reason behind this? Now, the people with AB blood group are the ones who on their RBC are having two type of antigen chains. That is, both the chains, the A chain as well as B chain, both of them are present on their RBCs. As a result of which, these individuals in their plasma, they are not having any antibody. So they will have no antibody in their plasma. Why? Because they have both the type of chains. They have A chain also as well as the B chain also. That is, they have A antigen as well as the B antigen. So in their plasma, there will be presence of no antibodies. This is the reason why the AB individuals are known as universal recipients. They can accept blood from any individual. Why? Because they are not having any antibody which is going to kill the antigens which are coming from the donor's blood. This is the reason behind AB being the universal recipients. So now let's see the options. Presence of antibodies, anti-A and anti-B on RBCs. So RBCs do not have antibodies. So obviously this option is wrong. The RBCs have antigens on them. Then absence of antibodies, anti-A and anti-B in the plasma. This is the correct option. Then absence of antigens A and B on the surface of RBCs. This is wrong because we've just seen that the A antigen as well as B antigen are very well present on the RBCs. Then absence of antigen A and B in the plasma. The plasma does not have antigen. Plasma has antibodies. So the answer to this question will be B, that is the absence of antibodies, anti-A and anti-B in the plasma. So these was another set of 10 questions which came in the NEET 2021 exam. Now in the next video, we will talk about the last set of 10 questions which were asked from zoology.